I shall now introduce KJ, who's been very tolerant. And KJ trained as a medic, but his research career until June this year was at the MRC Lab of Molecular Biology, and he's now moved to Oxford to be director of the MRC Weatherall Institute of Molecular Medicine there. We've been fantastically lucky with our opening and closing keynotes because both have FRSs for their contribution for the science, and KJ is also a fellow of the Academy of Medical Sciences. And what he investigates is toxic molecule formation from metabolism and how this might cause DNA damage and the way our bodies try to protect. But the focus for us today is on alcohol breakdown products and acetaldehyde in particular, and how that may contribute to the cause of some cancers. But I have to say, I first heard KJ speak about this when he was defending it extremely successfully as program grant, but I know he's a fantastic communicator of science, but he's absolutely not a killjoy. And in another world, if you wanted advice on a decent bottle of red wine, I'm well aware that he would be able to do that as well. So KJ, to set you up, thank you.
Okay, well, thank you, Ruth, very much for inviting me to give a talk in these very challenging times. We'll all be running to gin and tonics, irrespective of how much damage it might do to us at the end. Probably the only way we'll get some sanity in the current world we are in. So the story I'm going to tell you about, I hope everyone can hear me.
Yes, we can hear you.
So people are able to hear me, right? Yeah. So the story I'm going to tell you about begins with a very simple origins with a human genetic illness. And then how our study into this has led us to unusual connections to alcohol and other substances that metabolism generates. Probably what would be a good idea if the others mute so that I can't hear them.
They're already muted.
Okay. Maybe I'm the only one who's hearing them. So just to begin, what I want to introduce to you is a rare human genetic illness called Fanconi's anemia. And what happens to children with this illness is that they develop developmental defects. But what ultimately kills these individuals is progressive bone marrow failure. They stop producing blood. And if they survive this with a bone marrow transplant, well, then they go on to develop cancer. And the incidence of cancer is very, very high in these individuals. Just to put it into context, a child with Fanconi's anemia has a possibility of developing cancer leukemia about 10,000-fold compared to anyone else. So this genetic illness is caused by a very simple defect, and the defect is shown in the next slide. So the children get Fanconi's anemia and stop producing blood and get cancer because they cannot remove DNA crosslinks. Now, many of you are oncologists, so you're all very familiar what the DNA crosslink is. It's when the two strands of DNA are covalently linked together, and this is classically caused by the drug cisplatin. So in children with Fanconi's anemia, they cannot fix this particular block. And what happens is that when this block happens, it causes their chromosomes to shatter, particularly when they try and copy their DNA. And shown below here is an image of the chromosomes you get in Fanconi's anemia when the cells from these individuals are exposed to cisplatin. And you can see that these are highly abnormal chromosomes which are broken and fused together. So the main purpose of my talk is not a detailed genetic analysis of Fanconi's anemia, because this is a very extensively researched area, but it goes to the fundamental question about DNA crosslinks and where they come from. So this slide really sets out the key problem. You all know where a DNA crosslink comes from the kind of drugs you give to your patients. And shown here is a classic and most famous cross-linking agent, which is cisplatin, which glues the two strands of guanine, two strands of DNA through reacting to guanine. But when you think about it, an ancient system to remove DNA crosslinks could not have come into us simply to remove platinated DNA. There must be a natural substance that glues our two strands of DNA together. And that's why all organisms on Earth, right down to yeast, have a repair system. Actually, back to bacteria, have a repair system to remove crosslinks, okay? And so that is the main purpose of my talk. I'll first tell you what we believe is the factor that cross-links our DNA, and then give you some ideas of how we went out to prove that and what its implications are. So we first came to this discovery because we took cells from children with Fanconi's anemia and we exposed them in a dish experiment to a classical cross-linking agent, an agent that can cross-link DNA that's produced in us. And this is this molecule called acetaldehyde. So if you take acetaldehyde, and more, I'll tell you a little bit more about acetaldehyde in a short while, and you take cells with the Fanconi repair defect, you can see it rapidly kills them. So this is the first clue that we had that this simple molecule, acetaldehyde, might be the natural equivalent of cisplatinum that you and I produce every day. So this is a dish toxicology experiment. We wanted a physiological way to prove this. And so I turned to genetics and a clue I had in my previous life, which is as a hepatobiliary physician. So where does acetaldehyde come from? Well, our body produces acetaldehyde from many sources, but its most famous source is from that gin and tonic you had last night, or half a bottle of gin I had this morning while we were trying to get this started. So the body breaks down alcohol or body alcohols into acetaldehyde. And there's this key enzyme, ALDH2, that converts it into acetyl coenzyme A, right? So ALDH2 is the main detoxifying enzyme for acetaldehyde. So we did a very simple genetic test in mice to prove our hypothesis. What we did is we first deleted the ALDH2 enzyme in mice, and then these mice don't have a phenotype, and it'll become apparent to you. Many of you will have come across ALDH2 deficiency, and some of you may be afflicted with it. And I'll come back to that in a while. Now, if you now combine that with a mouse that lacks the DNA repair defect, you have a very simple genetic test here. You take away the enzyme that allows the cross-linking agent, acetaldehyde, to naturally accumulate in you. Now it comes along and cross-links DNA, and now you don't have the essential prop that prevents this, and you lack this repair system. And so what's the consequences? So just to reiterate, simple mouse genetics, we take a mouse that lacks the Fanconi repair system because it has a deletion in a key gene in the Fanconi pathway known as D2, and we combine it with a mouse that lacks this enzyme that lacks ALDH2, and by Mendelian inheritance, we get what is shown in red here is a mouse that lacks both protection systems, right? The enzyme that removes the toxin and the Fanconi pathway. Now, one thing I must tell you, and this caused a lot of controversy in the field, mice that lack the Fanconi repair system or cross-link repair don't seem to have any apparent phenotype. And this led to a lot of confusing thinking in the field, thinking that actually the Fanconi protein did something very unique in humans. And by the end of my talk, I hope I'll have dispelled that myth or that misunderstanding. So the key question is, what is the fate of the mice that lack the two-tier protection, these two critical genes, the enzyme that removes the toxin and the repair system? And shown in maroon here in a Kaplan-Meier curve is exactly what happens compared to the mice which are the allelic controls. What you can see is that these mice rapidly die. And what do they die of? Most of the mice die of acute leukemia. And that's very satisfactory for us, but not for the mice, obviously, because that is the disease that children with Fanconi's anemia get. They get acute leukemia. And the few mice that evade the leukemia all go on to develop bone marrow failure. So we have taken a situation where mice which lack the system to repair DNA crosslinks don't really have a phenotype, and just allowing acetaldehyde to accumulate will reveal all the features of the human disease in these mice. So that gave us strong confidence that acetaldehyde, this key factor, was the driving factor, was a cross-linking agent. But what you can see is this physiological experiment we've done doesn't really prove it is the acetaldehyde that does that. And this is where I went back to my penchant for alcohol. So what did we do? We did a very simple test, and that's to take and do an alcohol challenge in our sensitized mice, right? And so you can see the great famous Anglo-Saxon version of a human alcohol challenge. And the mouse version is a rather more boring thing, which is a bottle of alcohol or alcohol cut into their water supply. So what's the logic of this experiment? So remember I told you that ethanol is broken down into acetaldehyde. Now we take away this key enzyme that allows the acetaldehyde to accumulate. What I now need to tell you is that this defect is very common in man, because many of you will have colleagues who have what is known as the Southeast Asian flushing syndrome. And that's not an embarrassment syndrome. It is a syndrome whereby about 500 million Southeast Asians go bright red when they drink alcohol. And the reason they go bright red is that when they drink alcohol, they cannot break it down because they lack ALDH2. And the warning sign is it leads to accumulation of acetaldehydes that causes this flushing defect. Okay. So we went to our key mouse protocol, which we call the Costa del Sol protocol. And what is shown in this poster here is what the Costa del Sol protocol is, because most of us, before the COVID crisis really hit us, went to Spain for our holidays. And instead of drinking water, we go into a continuous beer diet. So this is what we did to our experimental mice, okay? And this protocol is shown over here, and I've called it the Côte d'Azur protocol over here, but it's the same thing. We take our mice and we give them 10 days of oral alcohol in their water. And then what we do is, at the end of that, we look at what happens. So in the wild-type mice, alcohol is very well tolerated, but in our double mutant mice that cannot clear the acetaldehyde and cannot repair DNA crosslinks, the bone marrow is completely wiped out. And this is a very, very striking finding, because by taking away acetaldehyde catabolism away and removing this DNA repair system, we have converted alcohol into a very, very potent cytotoxic drug, right? And it's doing this because it's causing DNA crosslinks in the cells that produce blood and causing it to ablate. So putting all this together, we came out with a very bold concept, which is known as two-tier protection. So all organisms have two tiers of protection against endogenous acetaldehyde or alcohol-derived acetaldehyde. Tier 1 protection is the enzyme that removes the toxin, and tier 2 protection is this DNA repair system. And just to say, deficiency in two-tier protection is actually very common in humans. So lack of tier 1 protection, as I've told you, is about 500 million people in the world. And these individuals we know are completely dependent on this DNA repair system, which is tier 2 protection. And tier 2 protection deficiency is Fanconi's anemia, right? Now, I haven't got time to tell you, but actually, if you go to Southeast Asia and you look at children who have Fanconi's anemia, but have also got this ALDH2 defect, the flushing defect, then these children have a very dramatic phenotype. They're born with bone marrow failure or acute leukemia. So all the features we've seen in mice are recapitulated in humans in this genetic setting. So two-tier protection, we believe, is real. So we then wanted to understand this a bit deeper. So remember, everything I've told you is that if you lack two-tier protection, you stop producing blood. And blood, as you know, is hierarchical, and most blood originates from the hematopoietic stem cell. So the key question we wanted to ask is, are the stem cells the ones that are impacted when you lack two-tier protection? And the question is, how? Now, why is this an important question? And it's an important question in cancer biology because of a very, very controversial paper published in 2014 from Bert Vogelstein that came out in Science rather cruelly just after Christmas. So just after we'd had our drinking binge after Christmas. And this paper basically argued, and it's a very simple hypothesis, the lifetime risk of cancer is not so much the factors you might be exposed to, but because of the number of stem cell divisions that your cells, that the cells that make up your organs have to undergo. And stem cell divisions is an important term because ultimately it means the turnover of stem cells and the mutations that they accumulate as a consequence of that wear and tear is what ultimately drives the majority of cancers. Now, it's not a view I subscribe to entirely, but why I was attracted to this hypothesis is because ostensibly in what I have described to you is a genetic situation where a firestorm of DNA damage from an environmental or an endogenous source is ravaging the production of blood, right? And we think this action is occurring at the level of stem cells. So we grasped this opportunity to prove whether the genomes of the stem cells are truly damaged in these mice, right? To prove whether an aspect of Vogelstein's famous conjecture was correct. So the logic of the experiment that I'm going to present to you, which took the best part of 4 years to complete, is the following. If you have DNA repair systems and you have damaged DNA, what DNA repair systems try and do is to completely fix that damage and make it normal, right? And so there's no mutations. However, occasionally, when a DNA repair system is lost, you damage the DNA, but then you put it back in a haphazard way because DNA repair is so essential for life, you try and get back to the status quo, but this then leaves a mutation scar. And if you are able to sequence and identify these mutation scars, you can then use mutational archaeology to infer the nature of the damage in the first place, okay? And so this is most famously done in the transformative work done at the Sanger from Mike Stratton and Peter Campbell. So how does this work? You will be aware that Mike's group, the Sanger, took the genomes of cancers and deep sequenced them and identified a mutation signature from them. And they could do this because a cancer cell, a lump of cancer, is an amplification of one genome, which then had mutations on top of it, right? What we wanted to do was to get the genome sequence of a single blood stem cell in our mice. And that's a bigger challenge because, as you can see, the bone marrow in our mice have very few of these cells. It's not like an amplified genome. So what we decided to do was to introduce clonality into the mouse, into a mouse model, so we're able to interrogate the sequence of a single stem cell. And we can do this because of a remarkable experiment done by a Japanese hematologist some 10 years ago. What they showed is that if you take a single blood stem cell from a mouse and park it in a lethally irradiated recipient, that single blood stem cell will then expand to produce a significant proportion of the blood in that mouse. And that means that if you then sequence the DNA from the blood that's originated from that single stem cell, you essentially have the same situation as shown at the top. You have somehow amplified the genome of the stem cell. Now, this idea, which is the idea of my graduate student, Juan Garacacheo, who's now a PI at the Hubrecht, I thought was a bit implausible, but we were at the LMB at that time, and it's a place where you can do this sort of experiment. So what did we do? This just shows you the big work plan. We took our mice, as shown in red, are the mice where the DNA damage is ravaging the stem cells. We took a single stem cell, visualized it, and transplanted it into a lethally irradiated recipient. And at the same time, obviously, asking a single stem cell to make all the blood would be an impossible task. You have to also give it a wild-type bone marrow transplant. But because you can genetically mark what comes from the single stem cell, what you can do when it's reconstituted is to sort the cells, the blood cells that have come from the single stem cell. And then we whole genome sequence that, right? And remarkably, this worked and allowed us to amplify the genome of the single stem cell. So what did it look like? So you'll all know these famous wagon wheel representations of the genome, right? Shown around in this circle are the chromosomes, and shown inner within the circles are the dots, the colored dots representing single point mutations that have been accumulated in the genome of a single wild-type hematopoietic stem cell. And the bars represent bigger changes, which are deletions. So the first thing you notice is that the stem cell of a mouse blood cell, a stem cell obtained from a mouse that is about 1 month old, already carries somatic mutations, which are the red dots. But considering the size of the genome, there are not that many of them. And thankfully, there are not many great changes, okay? Now, what happens in a genome, a stem cell genome that is being attacked viciously by acetaldehyde and also lacks this critical repair system? So shown here are 5 genomes from individual stem cells. And you will immediately see that the biggest change are the bars within the genomes, and also occasionally where there's an arc linking 2 chromosomes. So the take-home messages is if you accumulate acetaldehyde and you lack this repair system, ultimately what you get are stem cells which have a severely damaged genome with loss of genetic information and translocations occurring. And when we did this, this is the first time that the genome of a physiologically active stem cell had been sequenced to this extent, particularly in the context of a critical DNA repair deficiency and a common mutagenic driver. So ultimately what it told us is if you lack the Fanconi repair system and you cause a DNA crosslink, and what acetaldehyde does is glue the two strands of DNA, we think, the DNA snaps into two, and then you try and misrepair it and anneal it, and you lose genetic information. And what you get are these complex changes. But of course, the majority of stem cells die. Okay, so why is this important? I think it goes back to, if I go back to Vogelstein's conjecture, if your physiologically active key cell of regenerating your tissue begins life with a corrupted hard disk, shown at the top over here, then all its progeny are building on genetic changes that have already been subsequent to what has already happened, okay? So I do think Vogelstein's key conjecture is largely correct. Where I disagree with him is that it's the wear and tear of life, and that actually leads to the majority of cancer changes. If you have a mutagenic driver, either by tobacco smoke or alcohol, then it can accelerate this. And in this particular situation, we've been able to nail his conjecture and show it most strikingly. I haven't got the complete time to tell you about this, but the paper was published some years ago from Juan in Nature. But basically, it allowed us to show the entire pathway of what happens when you lack two-tier protection. Lack two-tier protection, your genomes of stem cells are damaged. The majority of these occur as double-strand breaks. And then you activate p53, which then kills the stem cells. Now, interestingly, when we took away p53 in our two-tier deficient stem cells, all the stem cells are rescued. But rather surprisingly, and reported in this paper, and completely unexpectedly, they did not wind up with massively scrambled genomes. And you will all know you don't lack p53, you great genome instability. And what Juan's paper, I think the piece of gold in his paper, because much of it is what you'd expect, is that in stem cells, if you lack p53, you allow a damaged stem cell to survive, but you don't pay a big penalty for genome instability. And that's truly a very surprising result. And we're very keen now to understand this better. And if you have questions, I might be able to expand on why we think this is the case. But it's well illustrated in this paper, and I won't go further into it. Okay, few stem cells escape the p53 response and repair and misrepair, and these are the ones we've captured in the deep sequencing, right? Now, if you have Fanconi's anemia, and you get bone marrow transplantation, virtually all the individuals are dying of head and neck cancers, right? So it's not unusual, and orogenital cancers, it's not unusual for 20-year-olds to have multiple oral squamous cell carcinomas of the mouth and anogenital cancers. And it's a very unusual spectrum which has been not explained before. So what we now did, and this is an experiment that's still ongoing, is we targeted two-tier protection just in the skin compartment, and asked what happens if we now just target it to this compartment in mice, okay? So first of all, the enzymes and the repair system exists in squamous cells. But if we do this, we engineer this remarkable finding in mice, which is shown at the top is a mouse that lacks two-tier protection in the skin, right? But if we give these mice 2 months of ethanol, this is what happens. The skin prematurely ages, and you can see completely loses the ability to produce hair. And I think this is important for two reasons. It may suggest an explanation of why oral cancers are occurring, because the aldehyde stress occurs in these occurs also in other tissues. But it also tells us that an important that discovery for for blood stem cells may be generalizable to other tissues like skin. Now, if you look very carefully, you will see that the tail of this mouse is black and the mouse has striking skin pigmentation. And we noticed this. And in fact, if you talk to Japanese dermatologists, many of them notice that their skin, their red flushes, if they go out in sunlight, get severely pigmented, right? And many of you may know anecdotally that if you give people chemotherapy, they get widespread melanotic responses. So we see this very strikingly in this animal model, and we're pretty sure what actually happens is very surprising. I haven't got the time to show the data. We think that DNA damage in skin squamous cells secretes a factor that tells melanocytes to secrete melanin. So the skin thinks that the DNA damage is coming from UV light, but of course it's coming from a natural source. So when you see a pigmented person after you've given them some of the drugs you give, maybe it's the same mechanism that's operating. So it just shows that animal models are very powerful to link desperate clinical observations. Disparate, not desperate, sorry. So I'm now going to go one step further, and because we have a reductionist philosophy at LMB and now at the Weatherall Institute that I've moved to as the director, I've taken to you a very compelling genetic argument about acetaldehyde causing DNA crosslinks and that requiring this repair system to fix it. And ultimately, I've shown you the genomic penalty of what happens when all this goes wrong. Now, the key piece of evidence is, can we actually show that if we make an acetaldehyde crosslink, that this repair system fixes it? Now, on the surface, this may sound a very straightforward question, but it isn't, because what we needed to do was to make an acetaldehyde GG crosslink. So I don't want you to get too caught up in the chemistry, but acetaldehyde causes a crosslink shown by this complex chemistry. And this happens in femtoseconds in our cells, okay? So Michael Hodgkinson in my lab, working in Jason Chin's lab at the LMB, we spent the good part of 5 years making a synthetic GG acetaldehyde crosslink, okay, in a duplex piece of DNA. And then we asked, how is this repaired? And we turned to a colleague of ours. So first of all, we made this GG crosslink. So if you look at this image here, what I'm showing you is a platinum GG crosslink rotating round, and next to it an acetaldehyde crosslink, okay? And without knowing much DNA chemistry, you will immediately recognize that the commonest model of a DNA crosslink is wrong. They're not the same. An acetaldehyde crosslink completely distorts a helix, right? It's almost unrecognizable as a double helix. Whereas an platinum crosslink, whereas an acetaldehyde crosslink shown on the right over here is very different. It very subtly alters across the DNA duplex. But obviously, if you want to copy DNA, you can't separate the two strands. So not all crosslinks are the same. So that's worth thinking, that all the cross-linking agents you might use in cancer treatment may differ in the way they distort the DNA helix. So the million-dollar question is, how is this crosslink repaired? So the go-to system is a frog egg system, because you need DNA replication to fix a crosslink. And so you need active DNA replication. So what Michael did was introduce the acetaldehyde crosslink into a plasmid, and then we flew it over to the Netherlands, where Puck Nipscher dumped it in frog extract and then tracked it, what happens. Now, I'm not going to bore you with the intense biochemistry of this, but what this analysis showed us is that a single acetaldehyde crosslink is repaired by two systems, and it was an unexpected result. The Fanconi repair system repairs it through one way by creating this repair intermediate and fixing it, shown on the left over here. But quite unexpectedly, we've stumbled on a completely new repair system that somehow dissolves the crosslink from within the DNA duplex without incising the DNA strands. And an important focus is to identify the enzyme that does this. And perhaps this is an enzyme that may work on the kind of chemotherapeutic agents you might work with, and might be a reason for resistance to some of them because it might be induced in certain situations. And you can see that this repair system does not create a strand break, so it does not use homologous recombination. So I think this is very exciting, but beware of a biochemical experiment that's detached from genetics, because we know that the main route must be this Fanconi repair system because of all the work I've told you already before. But that's going to be a point of interest in the future. Now I have a little bit more time, so I'm going to expand on my talk a bit further. But I hope the first part of my talk, the majority of it, you'll be pleased to note, I hope I've taken you to how our mouse model has gone from identifying a natural source and a constitutional source of endogenous DNA damage, how this action plays out in our blood stem cells. We've gone on to catalog how that damages the genome of blood stems, and now we've gone on to explain how, when we put that type of DNA damage into a defined piece of DNA, how that is fixed and what its consequences are. And all along has been a surprising array of collateral discoveries. And this work, or all this work, would not have been possible without the collaboration with Mike, Serena, Nick Zainal at the Sanger, and Pak Nipsch at the Hubrecht. Now, I've got a little bit of time to tell you that although acetaldehyde is what I've talked about with the Queen with her 4 o'clock, not 4 o'clock, maybe right now it might be the 4 o'clock gin and tonic. And on the right shown formaldehyde with Einstein's brain pickled in it. So you will all know formaldehyde because that is what you preserve tissues with, right? What you may not appreciate is that formaldehyde is the simplest aldehyde. It's the 1 carbon version, whereas acetaldehyde is the 2 carbon version. And actually, our body produces a lot of formaldehyde. So there are many biochemical reactions, like when you demethylate histones, that produces formaldehyde very close to DNA. Now, the enzyme that removes formaldehyde in us is a very conserved enzyme that goes all the way back to bacteria called alcohol dehydrogenase 5. And it's completely different from ALDH2, the enzyme I've talked about and told you about. And it does it through completely different chemistry and actually converts the formaldehyde to formate. Now, what we then went on to do is to ask, well, what happens if we remove the enzyme that clears formaldehyde in a mouse and combined it with the Fanconi DNA repair defect? So is there enough formaldehyde produced, at least in a mouse, to completely cook it if it lacked this protection? And what you can immediately see is that the mouse that lacks protection against formaldehyde is a dwarf, right? It's very small. And in the next slide, you will see that they rapidly die soon after birth. So, you know, as mammals, we produce a lot of formaldehyde in us, right? And if you shown in contrast of the green versus the red, the protection against acetaldehyde is much less important than the protection against formaldehyde, right? So formaldehyde is a fundamental fact of life. And prior to our work, nobody really thought of the fact that a pickling agent was omnipresent in our circulation. So all this really provides us with the following situation, which is that really there's a fundamental two-tier protection system that protects us from these 2 aldehydes. There's one that deals with formaldehyde in our blood stem cells, and there's one that deals with acetaldehyde. Now, I'll let you into an interesting surprise, and I don't know whether it'll be in my next slide. I've given you this simple formaldehyde, the 2 enzymes doing their separate things. Actually, if you make mice that lack the ALDH2 enzyme and the ADH5 enzyme, they're lethal. The mice are desperately sick, and very few make it into adulthood. And that tells us that the enzyme that removes acetaldehyde also does formaldehyde. And in a paper that should come out in Molecular Cell in the next few months, linking up with the Japanese group, we've been able to identify several families that carry that defect in Japan that lack the 2 enzymes. And all these children have severe myelodysplasia, bone marrow failure, and acute myeloid leukemia. So my simple concept of two-tier protection is a bit simplistic, because if you completely lack tier 1 protection against formaldehyde, you're completely cooked as a human being as well as a mouse. And it just underpins how much formaldehyde is produced in us. Now, I have a little bit of time, and I just want to quickly, and I'll whiz through some of these slides because I'm a bit behind time and I may have saturated you. But just to tell you that so far, I've talked about formaldehyde causing DNA crosslinks in this FA repair system, but the chemistry of formaldehyde should cause a multitude of lesions, which is a crosslink, a base adduct, a DNA protein crosslink. And those of you who do experimental work will know that you use this trick because you use formaldehyde when you want to do chromatin immunoprecipitation, right? Because it glues the protein onto DNA, and it also glues RNA to protein. So we believe that from the outset that there are other repair systems that were also important for formaldehyde, okay? And actually the repair system we think that's important should be nucleotide excision repair, which you'll be familiar with. And they come in two varieties of two limbs to it. Shown on the left is classical deficiency of nucleotide excision repair, which is xeroderma pigmentosum, this enormous predisposition to skin cancer. But there is a variant of nucleotide excision repair, which is really very, very fundamental in you and I, which deals with when you block DNA in transcribed DNA. Because if you don't relieve this, you can't transcribe a gene and you block the polymerase complex, the RNA polymerase II complex. And if you lack this repair system, you get something very unusual, which is children that are severely cachectic get neurodegeneration and develop kidney failure. This disease is known as Cockayne syndrome. And remarkably, these individuals are not cancer prone. Okay? So just to cut a long story short, it is formaldehyde matters for transcriptional couple repair, the Cockayne syndrome limb. And this simple experiment just shows that cells from mice that lack the Cockayne repair system are sensitized to formaldehyde. So the question is, can we do the same kind of genetic experiment we did with our Fanconi mice, with mice that lack the Cockayne syndrome? And it's an analogous genetic situation. The humans with this repair system are severely compromised, but the mice seem perfectly all right. And the question is, are they clearing the factor that's causing the damage much more efficiently than you and I are? So just to show that, the mice do not recapitulate Cockayne syndrome. However, if you take away formaldehyde catabolism in a Cockayne syndrome deficient mouse, or a mouse that lacks this repair system, they're severely growth retarded, shown in red over here. They develop neurodegeneration very rapidly, which is the cardinal feature of this disease. And the mice die of chronic kidney failure, and they get these small shriveled up kidneys, which actually is very similar to the kind of kidneys you get when you have cisplatinum toxicity, or the chronic version, not the acute version of cisplatinum toxicity. Now, what this tells us is, I'm going to move a couple of slides forward because I think this is a bit too detailed, and hopefully Nature will take this paper, but just really to get to the summary here. I hope I've convinced you, although I've told you there's a paper that will contradict my concept of two-tier protection, and it's good to have models that can be knocked down, that two-tier protection against formaldehyde is very important with this key enzyme ADH5. And if you do not remove the formaldehyde, you need this Fanconi repair system to make sure cells that are rapidly dividing in you and I are able to fix the crosslinks. And they probably use the copying mechanism, DNA replication, to fix it, okay? And that's the first part of my talk. However, formaldehyde will also cause the damage that will matter in tissues that don't divide, like kidney and the brain. And in those tissues, you don't care whether you put a crosslink in DNA that doesn't contain a gene. But if you put a crosslink in DNA that contains a gene, then you gum up transcription, right? And so I think the single endogenous driver has two critically dependent physiological repair systems to take care of the damage it causes in dividing cells as opposed to the damage it causes in non-dividing cells. And I think this is going to be a very exciting area for research for us in the future. I hope our paper will get published on the CSV bit because it has got one very unusual feature to it, which I can't help sharing my excitement about it. What we discovered briefly, and I haven't got time to show you the data, but next time I give a talk, I'll tell you, is the kidney failure occurs because the damage in the kidney occurs to cells in the proximal tubule, right? And if you give these cocaine syndrome mice cisplatinum, it is exactly the same cells that are damaged by cisplatinum. So repairing a crosslink in a proximal tubule cell matters. But remarkably, we discovered that one of the genes that is induced is a factor that causes the talks to the brain and gets you to go off food. So it leads to a profound food anorectic syndrome. And you may ask, why would you have such an axis? And I think the system was probably set up so that we knew that if we ate genotoxic food, it would be concentrated in the kidney, and the kidney would signal to make you averse to it, right? So that's just a hypothesis. But I think the cocaine syndrome part of the story on the surface may not be connectable to your world as oncologists, but I believe it's profoundly linked to it, because I think what we have discovered might in part explain the anorexia and the weight loss you get when you give people chemotherapy, which is you get DNA damage or crosslinks occurring in a cell in the kidney, a cell type in the kidney, which then secretes a factor that instructs your brain to go off food. So I hope this will get published soon and you'll be able to read about it. But it's been great fun in my research career. I should really get to the acknowledgement slide, sorry. To work with a brilliant bunch of people. I'm a very demanding guy, and they've really put up with a lot. And many of my colleagues have come to join me at the Weatherall, half of them at the Weatherall. Most of the key work done here was done by graduate students who've taken big risks to really push the boat out. And I think what they have discovered, not me, I've really provided them the tent and the funding for it, it has altered the field that we work in interesting ways, and I think has got clinical connections. And with that, I thank you for your patience, and we managed to survive, Ruth.
Yes, no, thank you, KJ. And one of the challenges of virtual platform is you can't hear the storm of applause, but that was absolutely fascinating. And, you know, a beautiful illustration of why DNA repair enzymes, as I would be bound to say, are really important. And, you know, but that using biochemistry and then these mouse models to tease it out. One of the things that occurred to me in the Asian flushing syndrome with the loss of ADH5, is that in effect protection enough to stop people who suffer from that testing the system with alcohol, or if they drink, are they more likely to get cancer?
Yeah, it's a very important question, and I you know, there's an epidemiological study buried in the literature about 20 years ago that showed that if you're an Asian flusher and you drink above 5 units of alcohol, your risk for oral cancers and esophageal cancers is high. So I have no doubt that the Asian flushers are putting themselves at risk with drinking alcohol. The good thing is that they feel so awful and they look so awful that they don't actually do it, right? So, you know, there's a natural protection system. And in the 2018 Nature paper, we take the ALDH2 knockout mouse, which is the correlate of the humans, of flushers, the 500 million, and then we give them a single dose of ethanol, and then we look at chromosome changes through exchanges. And the level of exchanges the mice get with a single binge drinking dose of ethanol is a bit akin to a dose of cisplatinum. So the message would be that a red flusher, when they go they light up like a pinball, are giving themselves the equivalent to their DNA to a dose of chemotherapy. Now, I'm sure people should really work on the flushing mutation. It's so interesting. So why is evolution selected for it? Why do 500 million people have it? It must be conferring an advantage. Then what are the consequences with it in the context of modern living? And we now know of 2 situations where it reveals itself catastrophically. If you lack the FA repair system and you're a flusher, you're completely finished, right? You're born with bone marrow failure at birth, and you'll get leukemia very soon after that. And now with the paper that's coming out in Molecular Cell, if you lack the enzyme that clears formaldehyde in that group, then you get Fanconi's anemia driven purely by a metabolic route. So who knows what other genomic changes puts this group into risk. And I think, it's a very long-winded way of answering your question, I think we'll see more of this because humans have never lived this long, right? You know, till about the 1930s, before the age of penicillin and after the age of major wars, mass extinction of people didn't happen. So happened frequently. So we now live with the cumulative damage that we put to ourselves at.
And with the clinical tragedy of Fanconi's anemia, presumably these patients are not actually getting to the point where they give themselves the double hit of exposure to alcohol.
No, they don't. But I came on this journey because of the paradigm of xeroderma pigmentosum, right? And I thought, well, look, xeroderma pigmentosum is a big problem because of sunlight. So, and sunblock mitigates against that. What's the sunblock equivalent of Fanconi's anemia? And what this journey has told us, ultimately, it is formaldehyde that's produced in us, right? Now, the big question is, can we cut off that supply of formaldehyde, take it away, a sunblock equivalent? And if we're able to do that, maybe we can improve the livelihood of these individuals. And that's something that we're very keen to work out. The story doesn't look very encouraging, though, because we discovered in a paper we published in Nature about 3 years ago that folic acid decomposes in us to produce the formaldehyde. So, unfortunately.
And we've got a question coming in saying, can we actually measure these enzyme levels to predict people who might be at higher risk of developing cancer?
The good news is assaying for the enzyme is not too difficult. But what we have done and invested a lot in, and it's shown in the Mole Cell paper, which will come out hopefully in a month or so's time, is how we can measure blood formaldehyde. Now, you'd think this is very easy. You know, we can measure sodium and creatinine and all that, but it took us into a 10-year journey to do this, right? And ultimately, and that's because formaldehyde is very small and highly volatile. And eventually, we found an atmospheric chemistry group in the University of Leicester that did it for the environment. And working with them, we developed a system to assay blood formaldehyde. And we come out with a very interesting number. In mice, it's very low. It's about 1 micromolar, which is still quite high. In a mice that lacks protection, it's about 5 to 6 micromolar, right? So it's 5 times more. In humans, it's about 5 micromolar, even with intact clearance. So that immediately explains why you get Fanconi's anemia, despite having the enzyme there, right? That clears it. Something about human physiology produces a lot more formaldehyde, 5 times more than mice. Maybe that's the reason, the puzzle of why mice that lack the repair system didn't have a phenotype. They didn't have the damage in the first place because the enzyme cleared it.
Now, there is a question coming that is pertinent because the government has recently changed the advice on alcohol in pregnancy and says that you should completely avoid it. Do you think, I mean, that's slightly outside your area.
No, it's bang on what we do. So in the 2011 Nature paper from Fred, we did a very critical experiment, which is we took mice that were sensitized, the 2-tier protection mice in utero, and we exposed the mothers that were carrying them to a single dose of ethanol. And we showed that the embryos were completely shot to pieces with multiple developmental defects. So we had a simple hypothesis that acetaldehyde could be a critical driver of the fetal alcohol syndrome. Now, I think this is a very desperate and sad disease because it's poorly studied and is ostensibly a condition of poor people, right, in the world. But when we think about, and we found the vulnerability is the first trimester, and it makes absolute sense. In the first trimester, right, the biomass of the mother is huge, but the fetal biomass is tiny. And now if a mother takes a bender in the first trimester, the amount of acetaldehyde you'd be subjecting that embryo and the few cells to is quite large if you think about it, right? Mm-hmm. So we think acetaldehyde drinking in the first trimester is a danger period. Now, cutting a full loop round to the story I told you about anorexia at the beginning, nature has a control mechanism. In the first trimester, you go off alcohol, right? Famously. The anorectic hormone, which I think is going to be very important for oncologists, and this field is just starting to emerge. And actually, the critical paper was published 3 years ago in Nature. It's a hormone called GDF15. I would urge you to go away and read that famous seminal paper that was published in Nature and Nature Medicine. There were about 5 papers from biotech companies. It's produced by cells that undergo DNA damage to secrete this factor. That's our work. But it's also produced by the placenta in the first trimester. And I think it's there because the minute you get fertilization, the first thing you say, Mum, don't go out there and forage for food because the food might be toxic to you. Hold back for a while and restrain yourself. And that's probably how I think physiologically what we've been studying can be united. And by the way, this hormone, this anorectic hormone, is probably the explanation for hyperemesis gravidarum. A beautiful paper published by Steve O'Reilly on this.
And absolutely, you're right. Off alcohol and sadly off coffee generally in the first trimester.
Yes. So there's a very interesting connection between anorectic syndromes and so forth, and DNA damage and constitutional dietary factors. And it makes perfect sense to use the kidney in the cocaine syndrome because the kidney is a great concentrator of crap, isn't it, of what's in our circulation, right?
But I mean, I know there's lots of work now on the anorexia and cachexia and how hugely important understanding that is for our patients. There's another question about which flips it to the other end of the spectrum, saying, does flushing with alcohol in postmenopausal women have anything to do with the aldehydes? Why does that?
No, it doesn't. But alcohol tolerance decreases with age. And many of us have grandparents or elderly relatives or friends who have very poor alcohol tolerance and stop drinking, right? Actually, I have an alcoholic uncle who underwent this transition, epiphany. And that is because there's an age-dependent decline in ALDH2 activity. So we all ultimately do become Asian flushers. How that happens and why that happens, we don't know.
I think on that sobering thought that we will all eventually stop enjoying our gins and tonics. All I could do is thank you so much.
It's a real pleasure, and thank you. And I know,
rounding this off in an absolutely fantastic way.